So, good morning, everyone. First of all, I would like to thank the organizers for inviting me to interact with you. Am I audible? So, we all are studying the different areas, right? So, you are in the pharmacy, I am in the biology, or I am in the biotechnology, or I am in the bioinformatics. But how we can correlate the pharmacy and the biotechnology, bioinformatics? So today everything is interdisciplinary and multidisciplinary. So every courses we need to come up with the multidisciplinary and interdisciplinary areas. So that says the NEP 2020. With the implementation of the NEP 2020, so we integrated with the computer science, statistics, and mathematics to solve the biological problems, right? So we will go through the today's my lecture of the data to discovery, the కొంచెం ఎక్కువ sound కొంచెం again. So now look at here what type of the data which we have. Then what we are going to discover. What type of the data we have, what we are going to discover. So in between how your bioinformatics is playing an important role. So that is what I am going to address today, right? So you look at here, when I say the bioinformatics is a interdisciplinary field that combines the biology, computer science. When I say the computer science, it is information technology or data science. So whenever you're having a data, the data is a huge data. So that huge data need to be classified. Huge data need to classify, need data to be organized. So that classification and organization of the data, you require a statistical and mathematical approaches. So for example, you need to do the mathematical studies, mean, median, mode, standard deviation, t-test, and ANOVA, MANOVA. So that require the statistical analysis. And you have a differential equations. So you have a computational fluid dynamics. So you require the mathematics. Are you operating? Babu, no operate చేస్తున్నావా? Is that? Why it is not going back? నేను హ్యాండిల్ చేసుకుంటాను, నువ్వు దాన్ని ఏం టచ్ చేయదు. ఓకే, నువ్వు కామ్గా కూర్చో, నేను చూసుకుంటాను. So now we have the mathematics and statistics. Whatever the data which we have, we need to analyze. So to analyze, and we need to interpret the data. So interpretation of the data. So, for example, take your phytochemicals. The phytochemicals you takes from the different sources. That may be a plant source, that may be a microorganism, that may be an animal source. Whenever you have a source, you need to analyze. The source is a biological material. So that biological material you need to analyze, right? So then, especially whenever you are dealing with genomics, whenever you are dealing with the proteomics, so you have a huge data sets with it. So that data sets, in terms of a sequence we used to take. So I can't directly go and take the human body. I will take the only human DNA sequence and protein sequence. So I need protein sequence, right? So whenever the bioinformatics is there, so tools that are developed to address the biological problems for the storing of the data, for analyzing the data, or perform some of the computation programs. So we have a commercial software as well as the private software, commercial as well as the public software. The public is nothing but open source software. Then, so it has many softwares, many algorithms that are freeware. So whatever the requirement which we have, so we can download and we can install and we can execute the program. So like that, the bioinformatics will place a important. So whenever you have a need, is it required bioinformatics today? Yes, bioinformatics is required. Bioinformatics plays a crucial role in advanced and HTS studies. So, for example, you have a genomics, you have a complex diseases. The complex diseases need to be identified. So, you need to study the human genome. So initially, the human genome understanding and analyzing the human genome, it started the HTTP project in the 1990. It is completed the 2000. So that means that 10 years time it has taken to do the genome sequencing of the human. So then only we are able to identify whenever you are affected with the disease, the disease is associated with the which gene, which protein we are in a position to say. But if your genome is not sequenced, if genome is not available, how we can say which chromosome is involved, which gene is involved, which localization is involved, which allelic position is involved. So that is the reason. So the analysis of the entire genomes, gene prediction, gene analysis, sometimes you need to compare, you need to annotate the gene, your genome with the another organism's genomes. We have many model organisms, that model organisms genome sequence also completed. So when I say model organisms, if I wanted to conduct the experiment, can I go and conduct the experiment in the elephant and dog, which are available in the local, or which are available in your campus premises? Then where are you going to conduct the experiments? That is the question. So the rodents are the animal models from the each species. For example, mammals. We have an animal model from the mammal is a mice and rabbit or guinea pigs. And if you have a bacteria and virus, we have an E. coli, yeast, and if it is a plant, proboscis. If it is a reptiles or if it is a arthropods, you have a Drosophila melanogaster. So like this, these are the model organisms. If you wanted to conduct the experiments on the fishes, so we have a zebra. So like this, some of the organisms, the genome sequence is completed. So whatever the way we wanted to conduct the experiments, we have to conduct the experiments on those animals or those organisms only. Because those genome sequence which we already know and how much we are similar, how much we are identical to that organism. So that is the reason. So we have required the genome and genome project. Then proteomics. So we have a gene and we have a protein. So the study of the proteins is the proteomics, right? So proteomics is studying the protein structure, protein properties, and protein function and protein interactions. The protein-protein interactions we used to say. So how the protein-protein will interact? Because from your head to your feet, so many proteins are in, so many genes are in. These all are interconnected with your nervous system, with the vascular system, right? అమ్మ నేను వచ్చింది నీకు రెండు ముక్కలు చెప్పడానికి మీరు మాట్లాడుకుంటారంటే మీరు మాట్లాడుకుంటే నీకు కాంబో రైట్ సో you have a gene and you have a protein. Then something is happened to your protein or something is happened to your gene. Then what the way we need to design a drug? The disease is in there. The disease is associated with the gene or disease is associated with the protein. Then the target is identified. That is called druggable target. Your drug has to work against the protein target or against your gene target. The gene is nothing but a DNA. The gene is nothing but a RNA. Then you have to design a drug. The drug design, so it is not readily available. So you need to synthesize the compound. You need to extract the compound or you need to isolate the compound, then you need to purify, you need to characterize, then you have to test against the receptors. Then receptor is not readily available, then you have to go and create. See, the disease is not there. We found a new disease. For example, breast cancer we found. Shall I take one madam and conduct the experiments against her? I will not take any girl or any madam to test my compounds. So what we used to do? Artificially we create a cancer, that is the breast cancer, to the animal. Then here we conduct the experiments. Then whether my drug is working, my drug is showing the activity or not, we used to check, right? So like that we used to check the drug design and development. First we will design the based on the activity, based on the receptor. Then we design the molecule in silico by using the bioinformatics methods. Then whether it is properly binding to the receptor, then we go for a experimental studies. Then, but paracetamol is working for me for reducing my fever. But the same paracetamol will not work for some others. Why? Because your body conditions, your physiological conditions, your adaptability may be different. So that is the reason today the person came to the picture. From your grandparents to you are going to only one doctor. That may be RMP doctor, that may be MBBS doctor, rather than you are going for MPR, the super specialty doctors. Because whether super specialty doctor is not curing your disease, because he is not properly understand your conditions, he is not properly understand your family background or family history, or which are your allergic, which antibiotics groups which work for your family. So, but immediately it may not be possible, but you course you can treat. But whoever treating your parents, you or your grandparents, he knows which antibiotic will work for your family. So whenever you go, so he will give. So right? So that is the personalized medicine. Just I'm simplified and told the answer. For personalized medicine, you require a higher intentional studies. For in-depth analysis, you need to know your genome, you need to know your metabolisms. Then we can design a particular drug for the individuals. But see how the bioinformatics is helping in the agriculture. So agriculture sector is a lot of the problems are there. That lot of the problems now being addressed by bioinformatics that we are saying that agri-informatics. Then, so we have a functional evolutionary biology and hybrid. So we all are the Telugu speaking persons, right? I say some comment. So your grandparents, maybe this Neelam and Sir knows, your grandparents may suppose when they are angry or when they are frustrated, kukkala kodka, nakala kodka or gaadi kodka అని చెప్తుంటారు. Have you heard? Because you are behaving like that. కుక్కలు అరుస్తుంటాయి కదా, అలానే నువ్వు కూడా అరుస్తున్నావేమో, అందుకే కుక్కల కొడక అని చెప్పారు. సో గాడి కొడక అంటారు. But they are not known, but it is somewhat, it is true. I am not comparing you with your donkey, I am not comparing you with the dog. But whenever you take your DNA sequence and dog DNA sequence, or whenever you take your genome sequence and donkey genome sequence, we are identical. But identical 95%, it's a 25%, 32%. So that is the reason we have the identity. The identity is what? In terms of the digestive system, in terms of the reproductive system, in terms of the way you are speaking, or vocal cords, circulatory system, vascular system, or whatever the legs we have, whatever the hands we have, the similar kind of a function the animals also will have. That's the way we are similar and identical. How I am saying it is because I have a sequence, I have a genome, then I am confident, right? So like that you can say. So when it is possible, you take the sequences and construct a tree. The tree is tree of evolution. The tree of evolution will be addressed and answered by the techniques. So today which we are using the clinical diagnostics, the clinical diagnostics are developed by the prediction of a druggable properties, drug properties for understanding genomes. So today we are generating the enormous data, but where do you store the data? Earlier, faculty members when they did the PhDs, so they used to study these many big literatures or big books. Today, you doesn't require any book. It's everything is available on the computer. Everything is available in a global platform. So the data which we are transformed into electronic. So whenever you have the electronic form, the data is available, then you will need to analyze. But to analyze the data, you require a software, you require algorithms. That algorithms which we are integrated, that algorithms are called machine learning algorithms, or algorithms are artificial intelligence algorithms. Right? So this biochemistry is not a single subject. It is an integration with the biochemistry, integration with the chemistry, integration with the physics, and integration with the biostatistics, and integration with the biophysics, and clinical biology, structural biology, and chemistry, that is called cheminformatics or cheminformatics. Right? So, what is the nature of the biology? So we have an expression of the biological data. We used to say gene expression, how the gene is expressed. You all are students. You all are students. But why 70% of students are calm and pay attention towards me? Why 30% of students are discussing? Why 30% of students are thinking something? Your genes are expressed in different way. Your actions are different way. Why you are not behaving like your friend? So the differential gene expression will be there in the condition to condition. So that we will study. So that we used to say the microarray. Microarray studies used to say about the gene expression properties. Then, So efficient of the data storage. So we are getting the data, whether it is a gene data, protein data, transcriptomic data, or where we have to store. So to store this kind of data, we require a software. We require a databases. So we have a sequence databases, structured databases, right? So structured databases and sequence databases which we are available. So if it is a DNA sequence databases, which we used to say the GenMap. If it is a protein sequence database, we used to say the UniProt, SysProt, etc. If it is a structure, we used to say that that is called PDB database. Then we have a complexity. So if I suddenly comes to you from here to there, you will be calm. You are discussing, you are making the noise. Whenever it suddenly comes to you, you will calm, right? Then who said to be calm whenever I suddenly come to you? I student will be set right. How it is happening? Because of body networks containing the 10 to the power of 18 networks that are called the nervous system is intercommunicated. I am speaking, you are seeing me, but you are thinking about your mother, your father, your husband, your boyfriend. You are physically here, but your brain is somewhere. Your actions are somewhere. So the human body is a complex mechanism. So we need to generate biological networks. Metabolomic studies we used to study. That is, we have a different pathways. So tomorrow your faculty member says the drug will work on a WNT pathway, JAK start pathway, or MAP10 pathway, P53 pathway. Every pathway is interconnected with the n number of the genes and n number of the proteins. That is the complexity of the biological system. One pathway is intercommunicated with another pathway is intercommunicated with another pathway. Otherwise, student, you have taken a diclofenac tablet. What is going to happen? Diclofenac or overend aceclofenac. మనకేం తెలియదు నేను చెప్తాను ఇక్కడ నొప్పి వచ్చింది ఇక్కడ నొప్పి వచ్చింది బాడీ జనరల్ అనాలిసిస్ ఉన్నాయి మీరు ఇచ్చే అసెక్లఫినాక్ డైక్లోఫినాక్ అక్కడ పనిచేస్తుంది మరి అది వాడుతున్నప్పుడు ఏమవుతుంది కడుపులో అల్సర్స్ ఎసిడిటీ జనరేటర్ దాన్ని తగ్గించడానికి ఏం చేస్తాం ప్యాంటాప్రజోల్ ఇస్తాం ఆర్ ఇసంప్రజోల్ ఇస్తాం ఆర్ ట్రాంటాక్ ఇస్తాం సో ఒకటి ఇక్కడ డ్రగ్ తీసుకున్నప్పుడు అది ఇక్కడ ఎలా పనిచేస్తుంది ఇక్కడ ఎలా క్యూర్ చేస్తుంది ఇక్కడ ఎలా క్యూర్ చేస్తుంది నువ్వు నాప్రాక్సిన్ టాబ్లెట్ తీసుకున్నావు మీకు చాలా మంది మైగ్రేన్ ఉంటుంది ఇక్కడ హెడేక్ వస్తుంది కానీ టాబ్లెట్ ఏమో ఇక్కడ తీసుకుంటుంది మరి ఇక్కడ ఎలా పనిచేస్తుంది సో డ్రగ్ విల్ ఎంటర్ ఇన్ టు యువర్ మెటబాలిజం keep in mind directly your capsule will not come to here and will reduce tablet pill will not come to here and will not reduce that will dissolve that first dissolves then it will enter into the metabolism right so personalized medicine and so this is a drug discovery and development so then so genome containing the gene is translated into a protein or transcribed into a protein. Then this DNA is transcribed into RNA, RNA is translated into a protein. So when you have a DNA to RNA, RNA to protein, what we can say? So make the logo it is a central dogma of the molecular biology. DNA to RNA, RNA to protein, it's a central dogma, right? So this we will, whatever the studies which we conduct, so that is completely associated with the genome to protein levels, right? So whenever you are taking any DNA sample, or protein samples. We have a qualitative studies and quantitative studies, right? The qualitative studies and quantitative studies. So whether you are interested the quantity, so your spectrophotometers or some of the studies, they used to see the quantity of the products, or your chromatographic studies will also see quantitative studies. Then to have a quality of the studies, gel electrophores or PCRs or gene expression studies, etc. will talk about your qualitative or quantitative and qualitative study. Then I have a DNA. How can I access? So we have a different methods. One way is the use spectrophotometer, another way is the fluorimeter, and we have a perchloric acid test. Maybe students are doing this basic test, that is called perchloric acid method, or ethidium bromide test. Then you may be seeing the agarose gel electrophoresis and picogreen assay and diphenylamine method and offset method and DNA slab blood analysis. So these are the methods that are available for the total DNA estimation from the whatever the sample which we have taken, right? So then, so you have another method. This is the laboratory condition where the graduate students are there. We used to conduct that experiments in the degree level and PG level, right? But I have a sample. I need to extract from the DNA. I need to extract the DNA from that sample. So how can I do? So we have the Maxwell Gilbert method. So we have an enzymatic method and we have a chemical method. So that is a Maxwell Gilbert method and Chancellor method, light sequencing, automated sequencing, large scale sequencing strategies. So when you have a large sequences, so we have a short reads and long reads will be there. So how these long reads and short reads will be there? Advanced methods are called the next generation sequencing methods are there. So these next generation sequencing methods, whenever you have a large sequences, it will be fragmented in the short sequences by using the NGS protocols. Then, so whenever you have a sequence or whenever you do the sequence, then we have a DNA or RNA or protein. But it has some part. So you have a videos. What are the format it will be there? Either AVI format or MP4 format. You have a songs that is the MP3 format will be there. So you have a JPG, your photos will be there in a MP JPEG or PNG format. Here also, whatever the sequence I have, that sequence is also in a some specific format. So that may be ML format, that will be FASTA format, DCG format, and GenBank format, or AG format, and flat format will be there. So it will be varying from the database to the database. Then, so the similarly we have a RNA estimations. So these are the complete RNA